different chemicals in order to prepare the micronutrients. I will show in the next slide. Okay, so for the media preparation, we won't add. Hello. So we won't add all the chemicals at a time and mix up all the chemicals. So what will happen? It won't get dissolved. So what we have to do, first we need to add one chemical, then we need to dissolve using magnetic stirrer. Once it gets dissolved completely, then only we will add another chemical. Like that way we have to prepare the micronutrients, macronutrients and vitamins. So we need, for that preparation and all, we need magnetic stirrer. And what and all we need? pH meter. It plays a very important role. Without pH we can't prepare a media. We can prepare a media, but pH should be maintained. pH plays a very important role. You know what is pH? Power of hydrogen ions. So we need to maintain the pH in the media. So what will happen if you want to maintain the pH levels? There is no growth of plant. There is no development. We can't see the development in the plants. Okay. So for checking the pH, we need pH meter. Then what and all we need? Autoclave for sterilization and hot air oven. These are all fridge to store the chemicals. Some chemicals will be there, some hormones that we cannot store in normal temperature, room temperature. So we have to store in the fridge. So we need one fridge also. These and all we will arrange in the working room. So after working room, we so next followed by మనకి ఏముండాలంటే sterilization room. So once మన media prepared చేసిన తర్వాత we need to sterilize. Sterilization, it plays a very important role. Next slide I will tell what are the sterilizations, how we will do that and all. It is there in the. So we need one sterilization room. So after that followed by washing room should be there. So if once the medium gets contaminated, or if you want to wash the bottles or conical flask or whatever it may be, so we need a washing room. Then this and all should be one row. And next side we have to establish inoculation room, that we call as inoculation chamber. So in inoculation chamber, what and all should be there? What and all should be there, ma'am? Laminar airflow. Laminar airflow chamber. Okay. So only laminar airflow chamber will be there. Inside the laminar airflow, we have some equipments like surgical plates, tissue papers, cotton, ethanol, and forceps. That plays a very important role. If forceps is not there, how we will place the material? Right now? So we need forceps also. Okay, after that, once we get the inoculation part, another room should be there, that is followed to the inoculation room. That is called incubation room, that we call as incubation room. What and all will be there in the incubation room? Hot air oven. No. Culture rack should be there. Growth chamber. So that should be, the second two rooms, what I call inoculation room and the incubator room, that both rooms should follow the biosafety 2 or 3 we have to maintain. Why? Because that should be so sterile, it should maintain perfect sterile conditions. If we are not maintaining the sterile conditions, definitely whatever the work we are doing, that will become waste. So we have to maintain biosafety level 2 or 3. The biosafety level 1, it will be maintained in the first rooms, which I said that media preparation room, sterilization room. It is a public traffic where we can go and we can run off from that. But when we are entering into the inoculation chamber or the culture room, we have to maintain. See, when we are entering also, we have to wear a mask, head mask, mouth mask, head mask, gloves, this and all aprons you have to wear. and you have to enter. If not, don't enter. It is not like a public traffic. ఏదో అంటే అలా వెళ్ళేసి అలా రావడం అలా ఉండదు. ఓకే, సో ఓకే, ఇనాక్యులేషన్ సో ఇంక్యుబేటర్ రూమ్ లో ఏమేమి ఉంటుంది? కల్చర్ ట్రాక్స్ ఉంటుంది. ఓకే, గ్రోత్ ఛాంబర్ ఉంటుంది. సో వాట్ అండ్ ఆల్ ద కండిషన్ వి విల్ మెయింటైన్ దేర్ ఇన్ ద ప్లాంట్ గ్రోత్ ఛాంబర్? aseptic environment. Next thing, low temperature. Cultural conditions. What are the conditions generally we will maintain, ma? So temperature. So temperature, how much temperature we will maintain? 25 degrees. 2 to 25 degrees, they say 25 degrees we will maintain. In light, photo period, that plays a very important role. So oka plant grow avalante, these and all it will need, and we will maintain the humidity. ఒకవేళ మన హ్యూమిడిటీ ప్లేస్ చేయమైనా పర్వాలేదు, బట్ దీస్ టూ ప్లేస్ ఏ వెరీ ఇంపార్టెంట్ రోల్ ఫర్ ద ప్లాంట్ గ్రోత్. ఎందుకంటే మనం ఆ గ్రోత్ ఛాంబర్లో పెట్టేటప్పుడు మనం లైట్ ఎలా ప్రొవైడ్ చేస్తాం? బయట అయితే నార్మల్గా మనకి సన్లైట్ వస్తుంది, ప్లాంట్స్ అవి యూజ్ చేసుకొని దే విల్ గ్రో నైస్లీ. బట్ when we are growing in a chamber, this and all we will maintain. So light photo period ఎలా మెయింటైన్ చేయాలి? 16 hours photo period, 8 hours darkness. సో ఇనిషియల్ గా సీడ్ జర్మినేషన్ కి అయితే 1 week మనం డార్క్ కండిషన్స్ లో ఉంచుతాం అన్నమాట. సో 1 week క్యాలస్ ఇనిషియేషన్ లో కూడా అంతే. సో 1 week డార్క్ కండిషన్స్ ప్రొవైడ్ చేసిన తర్వాత దెన్ we will provide 16 hours photo period and 8 hours darkness. So these are the lab facilities, general lab facilities, these and all. So you understand, right? So okay, next. Okay, ma. So once we start to plan to work on plant tissue culture. Now lab is ready. So what and all we required, we have arranged the lab. Then next what? We need glassware. Without glassware, how we will prepare the media? Right now? So we need to procure the glassware. So what and all the glasswares and what and all the materials? So the glasswares like conical flask, bottles, culture bottles, and beakers, measuring cylinder, these and all comes under glassware. Now chemicals. What are the chemicals we need? Micronutrients. Magnesium, manganese, zinc, iron, molybdenum, sodium molybdenum, copper sulfate, ammonium sulfate. This and all comes under micronutrients. And what are the vitamins? Generally we will use vitamins: thiamine, nicotinic acid, pyridoxine. These are the vitamins we will use. Carbon source, sucrose. Amino acid, glycine. Very good, very good. You people are very attentive and you are answering very well, very good. Okay, so these and all chemicals we need, right now? So these and all the chemicals we will need. Then next one, surgical blade. If surgical blade is not there, how we will cut the explant? We can't cut now with scissors and all. We can't cut because see, when we want to generate a callus, we have to side part of leaf and all we have to cut like this. The outer part, the edges we need to cut. The injured part only will use the proliferation of cells. If not, if you just cut and put, then you will get a very little callus. But when we injure the part completely, little better, then only we will get mass of cells protruded from that explant. So we need surgical bed. Then forceps. If forceps is not there, we can't inoculate. So we need forceps, right? So next, Petri plate. Why I am saying Petri plate in this? Because see, if you take an explant, where we will keep that explant and we will cut on the bench? No. We need some Petri plate or we need some glass plate. Okay now. So we will wipe with 70% ethanol. Then we will keep the explant. Then we will excise nicely. Okay now. So next one, cotton rolls. So why I told you cotton rolls and all? Cotton flex. To wipe the laminar air flow and to wipe all the instrument which we are using inside the laminar air flow. So we need cotton. Okay now. So tissue papers. Why I mentioned here tissue papers? After surface sterilization, we need to remove the water droplets on the seeds or the explants. So after sterilization, immediately we will transfer inoculate. So we need tissue papers. This and all the basic requirements we need. Materials. Okay, sterilization. I told you sterilization plays a very important role. Well, see, we are working on microbial cultures. In what is how many days we will get the result, ma?
3 days.
So suppose I am saying if you are you prepared a broth and if you inoculate an organism, just E. coli, streptococcus or whatever it may be. So how many days we will get the result? In how many days? Overnight. 1 day is enough for multiplication, getting a good volume of bacteria. Right now? But whereas for plant tissue culture, it depends on the plant. It will come, the germination